This week in Phages, we are going to be covering our first research paper. And this research paper is titled, Bacteriophages as an Alternative to Conventional Antibiotic Use for the Prevention or Treatment of Paenibacillus larvae in Honeybee Hives. So this is a short introduction video that will give you everything you need to know to start reading this paper. So first, here's some tips on reading a scientific research paper. First, here are some things that you definitely should do. You should be appropriately skeptical. And what do I mean by appropriately? Well, you should be skeptical, but you should also trust that the authors of the paper have done their best to report the data accurately. Right? You should trust that the peer reviewers have done their job to carefully review the paper. But you should also recognize that people who have spent months of their lives on a particular study probably have biases in favor of their particular hypothesis. And you should recognize that even in the results, and especially in the discussion, there are interpretations of the data. And you should read the data because that is what a scientific paper is there for. In order to do that, you're going to need to take your time. Right? There's in every scientific paper, there are a lot of dense, jargony terms, and you're going to need to look up terms that you're unfamiliar with. Right? This is something I have to do every single time I read a paper. There are lots of words in there that even I don't know, and it takes a little bit of time to work through some of the dense language that people, especially working on projects that I'm not completely familiar with, have used to describe their work. The more practice you get with this, the better you're going to be at it. And I hope that you all are pretty comfortable by the time we're done with the semester. So the other thing you're going to have to take time to do is understand how and why the experiments were done. Right? So if you want to interpret the data, you have to understand something of the methodology behind those experiments. It's also pretty helpful to think about how the experiments were physically done. Right? How difficult is it to pipette 256 samples into a micro-titer dish, for example. So like I said, there are many different ways that you can read the paper. Here are my particular strategies. First, I like to read the abstract. So the abstract is a brief synopsis of what's in the paper. It's a good idea to go through the abstract, read it carefully, but circle and look up some terms that you don't know. I frequently use Wikipedia, or if it's just a regular dictionary word that I don't know, I use dictionary.com. Right. At the end of the abstract, you should be able to restate what the paper is about out loud to yourself. And this is a helpful exercise to make sure that you really understand what the authors are getting at, or the gist of the paper, before you dive into the details. Right. My first read through the paper, I always just I ignore all the text other than the abstract and look just at the figures. I do read the captions for the figures, because those will tell you what the figures are about. And so I just spend time looking at the figures, looking at what the labels on the graph's axes are, what the labels on the table heads are, just to understand what each one of those data figures is presenting about. I don't need to understand the experiment entirely at that point, but I ask myself a lot of questions about what kind of data is being presented in this figure and why are they doing it. After I do that, I go back and read the paper. I read the intro, results, and conclusions, and during that time I will pay attention and do a second read of the figures, which are usually heavily referred to within the results section. After I have read those parts of the paper, then I read the methods section. The methods section is usually a very technical and dense section of the paper. It often includes a lot of methods and things that I don't know. The methods in a scientific paper are written in very brief shorthand. They're not a detailed step-by-step protocol. And so it can sometimes be a little bit difficult, even for me, to figure out what those methods are explaining. So I take my time in reading methods to try and carefully figure out how each experiment was done, and I do that in line with a third reading of the figures, so that I'm thinking as I read the methods how was the data on this figure generated? So at the end of this process, I have carefully looked at the figures 3 times. And again, we're really focusing carefully on the data and on those figures. So this is the abstract of the paper that we're reading this week, and I thought it might be helpful to go through this and just talk you through the things that I saw when I first looked at this paper. First, there are a number of terms on here that were technical, but that I knew what they were, and so they're circled here in green. So hopefully you're all familiar with these terms as well. By now you have described to me what a bacteriophage is, right? We also hopefully are familiar with the common honeybee and that they live in honeybee hives, right? But we'll talk a little bit more about why those are important. And also, you might be familiar with the term antibiotics. So antibiotics are drugs that we use to treat bacterial infections. I also saw in here a number of terms that I was not familiar with. And so I immediately circled those and I said, these are things that I think I should look up. Right? Oh, so Panibacillus larvae. So I did not know anything about this bacteria. I did, however, recognize that this was a bacteria scientific name because it uses this binomial nomenclature formatting. Right? So I first looked at this, and it says in the first sentence here in the abstract that Panibacillus larvae is a bacteria that causes the infectious disease American foul brood, which we'll also talk about in here. Right? So I took the opportunity, though, to look this bacteria up in Wikipedia, and I'll tell you a little bit about what I found. But I recommend you do that as well. There are a couple other terms in here. In the article info, there are a couple of keywords that are things that are important in the paper. Tylen was a name that I was not familiar with. I looked that up, and that is a particular type of antibiotic that we'll talk about too. Then there was this term in the abstract where they measured something by a Hitchcock score, which was something that I had never heard about either. This seems to be a technical scoring method that many papers use different methodologies to measure things, and here the Hitchcock score is measuring the health of the beehive. So these are sometimes things that are not well spelled out in paper, but luckily in this particular paper, in the methodology section, which I hope you go through, the Hitchcock score and what it measures is pretty well defined within that. And so that was very helpful, something I liked about this paper. Then I went through this abstract and I highlighted some claims that I think that the authors were trying to make, some results that they were highlighting in support of their bacteriophages as an alternative to conventional antibiotics. Right, so in that first sentence, they're saying that these bacteriophages are an alternative, and hopefully they present some data supporting that. They make a number of claims in this abstract, which I've underlined here. First, they say that the phage cocktail did not adversely affect the rate of bee death. Right, so that should be something that we think about in the paper itself. Then they also make a number of statistical claims. For one, they say hives treated with phage cocktail decreased 19 plus or minus 0.08%, which was not statistically different than control hives. Right? Which decreased 10 plus or minus 1%. And so that is a claim that seems to indicate that these two things are not particularly different, but the numbers do say that they are somewhat different by what they measured them. And so that's something that we're going to have to delve into just a little bit. Because one of those numbers is 19 and one of them is 10. Right. The last thing that I highlighted in here was that the authors claim that for both the phage protection rate and the phage cure rate, those were statistically significant. Right? And those are claims that the authors are making that this phage intervention was effective at protecting and curing these hives. And so that seems to be the root of the claims these authors are making in the paper. And so those are things that we're going to look for in the data and say, does this data support the claims that these authors are making? So why do we care about this paper? For one, honeybees are super important for U.S. agriculture and for agriculture worldwide. Those bees are not just making honey. Bees are the primary pollinators of all kinds of crops and most of the food that we eat. Right? It's estimated in one report I read that bees and their use in agriculture adds $8 to $14 billion annually to U.S. farm production. Right? So on the right-hand side here, there's a commercial bee truck. So these pollinating bee operations are actually a really big business. So these bee colonies that are as big as a semi-truck are trucked all across the country as the seasons change in order to pollinate the different crops that are flowering at that time of year. So oftentimes these trucks will go from California in the spring when the almond trees bloom all the way up to North Dakota to pollinate some of the wheat fields. And all the way to Maine to pollinate the blueberries. And so these trucks are carrying these bees around the country. They are getting paid big money by farmers in order to make sure that their crops are well pollinated. And this is a major industry in the United States. So these bee colonies, however, are threatened. So these bees are sensitive to a number of things, including chemical insecticides that we use to protect crops from other insects, and also a number of insect pathogens, including parasitic mites, viruses, and bacterial illness. One of the biggest bacterial illnesses, it seems to be, is a disease called American foul brood. So this is a disease that affects honeybees, and it's caused by the bacterium P. larvae. Right? So Paenibacillus larvae, and don't worry about pronouncing these words. I am not even 100% sure I'm pronouncing this word correctly. I'm pretty familiar with bacillus, but other than that, I'm just making it up as I go along. Right? But P. larvae, I'm going with that, is a gram-positive rod-shaped bacteria. Right? So it is shown in figure A over here on the right. So it's a spore-forming bacteria. So spore-forming bacteria are capable of producing this protective protein coat that allows the bacteria to survive harsh conditions for long periods of time. And this is important for this bacteria because honeybee honey is naturally antimicrobial. And a lot of bacteria don't survive and reproduce well with some of the chemicals that are in the honey. Right? So you'll notice that honey that you keep in your at room temperature in your cabinet is good for many, many years, and that's because that honey has some natural antibiotic properties, and it doesn't allow spoilage by allowing bacteria to grow. But these spore-forming bacteria are able to transport themselves inside of the honeybee honey gut and therefore spread around the hives. So in this paper, they discuss several strains of this P. larvae bacteria. So strains are genetic variants of the same bacterial species. So remember, the idea of species is a little bit different in bacteria than it is in animals, because all of the bacteria reproduce asexually, and so those bacteria generally have their genome copied identically every single time they divide. And so the bacteria populations have small differences between them. So even some bacteria that behave pretty differently can have small genetic variations. They're not different enough to become different species, but they are different enough to behave differently. And some examples that you might be familiar with are the bacteria Staphylococcus aureus, which causes staph infections, and its strain variant, methicillin-resistant Staphylococcus aureus, which is or MRSA, which is a variant of that bacteria that is resistant to some of the antibiotics we use to treat those infections. We'll also talk about E. coli, which is a non-pathogenic bacteria that lives in all of your lower intestines, and enteric toxin producing E. coli, or ETEC. And this is a pathogenic version of E. coli that you might be familiar with from a number of foodborne illness outbreaks. One interesting thing that I did find when I was researching this P. larvae bacteria, so this figure on the right is actually from a CDC review where they found infections of this bacteria in humans who had been using intravenous drugs, so they had been injecting methadone that was laced with honey. And so there were 5 cases of human infections of this spore-forming bacteria. So, as an aside, it seems like not a good idea of something to do. Antibiotics have widespread use in agriculture around the world. So antibiotics are chemicals that kill or inhibit the growth of bacteria. So antibiotics work in a number of different ways, but they all generally inhibit critical biochemical pathways, including things like ribosomes, which make proteins, and the synthesis of bacteria cell walls. So the really crucial structures for bacteria, the antibiotics stop from working. So antibiotics are really effective medicines, and we use them to keep infectious bacteria at bay. They're very effective at curing a number of bacterial pathogens. Right? They do have some downsides in medicine, so they do also kill off good bacteria. And so oftentimes a number of antibiotics have the side effect of causing diarrhea or stomach discontent because they are not only killing the pathogenic bacteria that you're taking the course of antibiotics for, but they also kill out a number of the good bacteria that are naturally living in your gut, and that can cause some dysregulation. One problem with antibiotics especially is that antibiotic resistance among bacteria who are resistant, who aren't affected by those medical antibiotics, are becoming more prevalent. And one of the reasons is that we also use antibiotics widely within agriculture. So we have a number of bacterial pathogens that cause damage both to honeybees and livestock animals and also to plants. And we use antibiotics in agriculture to stop these diseases. In some cases, we even directly spread those antibiotics on crops to prevent bacterial diseases in plants. Those antibiotics are widespread in animal feed. So they're put into animal feed because they promote the growth, promote the weight gain of those animals, especially in cattle. Right? Tylen or tylosin is an antibiotic widely used in agriculture. Right? On the right-hand side here is a picture of Stuart Levy. So he was one of the researchers who first raised the alarm that the use of antibiotics in agriculture was leading to the spread of antibiotic resistance. In 1976, he published a paper that showed that antibiotic resistance genes, when antibiotics were used in agriculture, were rapidly spread from chickens to other chickens and then into farm workers. Right? And there's a description of this study in the book by Marilyn McKenna called Big Chicken that you can read an excerpt of on Wired.com. So antibiotics are really useful medicines. So there are some, but there are also, we need new ones. We need new options for antibiotics. And phages promise to be a possible alternative to antibiotics. So there are pros and cons of both of these things. So antibiotics, some of the pros is that they are effective against many bacteria. And that they are also pretty cheap to manufacture. Some of the cons is that they are also effective against many bacteria. And so those antibiotics will kill many bad bacteria, and they will also kill many good bacteria. They also, because they affect so many different bacteria, they can lead to the evolution of resistance in lots of different bacteria that can then spread among populations. So we've seen widespread evolution of antibiotic resistance in recent years, but at the same time, the development of these new drugs has slowed. Right? And so we're looking for alternatives to use to these wonderful medicines. So phages, because they kill bacteria, could be a possible alternative to these antibiotics. Phages have a lot of advantages. For one, they only affect specific bacteria, We talked about the host range. They're very cheap to discover too, right? We can do that process in this class here. And we've discovered lots and lots of phages just through the Z-Phages undergraduate research course. Right? There are some cons about phages. For one, they only affect specific bacteria. Right? So just like this is that antibiotics are effective against many bacteria as a pro and a con for antibiotics, phages, they only affect specific bacteria, and so that makes them very specific. However, if we want to treat a wide variety of different strains or different infections, we need to isolate new phages every single time, unless we find phages that are widely effective against a bunch of different bacteria, which is not very common. The phages also tend to be a little bit expensive to manufacture. We don't have all of the industry for producing lots of phages, and especially since we need to figure out a new one to use in lots of different cases. So because of this, and because antibiotics have been widely effective for a long period of time, phages are not traditionally used in the United States. Phages had been in widespread use in the Soviet Union in the 1980s. There was a lot of research on using these as medicines in the USSR, and they're still commonly used in some of those former Soviet bloc countries. But in the US, this is a relatively new intervention that we're looking at. So the approach that the authors take in this paper is very similar to the approach that we are taking in the phage discovery course. Right? We are isolating bacteria by collecting environmental samples and then using those samples to find phages that infect a specific bacteria that we chose in the lab. Okay, so we're isolating phages on, in our case, Microbacterium foliarum, but in the paper's case, they're isolating phages on P. larvae, so they're using that as a host organism. Right? They go through the same process of scaling up their phages, and then once they have characterized and found a whole bunch of phages that are effective against their larvae, their P. larvae bacteria, they do another assay that is part of the phage discovery guide protocol book. So it's one of the extra experiments that some students do at the end of the course. It's in protocol 11.5 in your phage discovery guide. This is a host range assay, and it uses a lot of the similar techniques that we've learned about in this most recent unit. So this assay's goal is to test to see if a phage isolated on one host can infect other bacteria. So to do this, we take a few easy steps. We make serial dilutions. So you make a serial dilution of a phage that you know infects one bacteria. And then you spot those different dilutions on plates of different bacteria. And then observe the extent of clearing. So on the lower right-hand side is actually a host range assay from the next paper that we're going to cover in this course. And in this case, this phage called Muddy was isolated on the bacterium on the right, the M. smegmatis MC squared 155. And they made serial dilutions of this one phage, the same lysate, and spotted those dilutions on 3 different plates. One of those plates had the original host bacteria. And you can see as those serial dilutions go down, the extent of clearing also goes down. You can see in the middle that they spotted it on a strain of bacteria called M. abscessus GD01, and you can see there's decreasing spots on that as well. And then on the third one, you also see an M. abscessus, a different strain, GG03, and you see a little bit of clearing on the very left-hand side, but not a whole lot on that assay. Now, I'll remind you that the exact same phage samples, the same serial dilutions, were spotted on each one of these plates. Right? So the phages are the same on the exact same thing. Why is the pattern of clearing different? Also, think about this phage was originally isolated on M-smeg, MC squared 155. You can see clearing on that plate down to about the 5th dilution that they've plated here. On the middle plate on this other bacteria, you can see clearing all the way down to the 7th or 8th dilution on that plate. And on the plate on the right, you don't see a whole lot of clearing except maybe on the highest concentration on that plate. So which one of these bacteria do you think is more susceptible to this particular phage muddy? See if you can spend a little bit of time analyzing that data. One of the tools we use when we analyze data are statistics, because many of the experiments that we do in the lab involve some amount of either randomness or variance. And that randomness or variance can arise from a number of different sources. Sometimes we make minor measurement errors. If we're using a pipette, we maybe don't have the most steady hand and make minor variations in the volume we pipette every single time. We often make mistakes, or the way we repeat things is not 100% the same every single time. Statistics are a mathematical way that help us quantify how much variation there is in a set of data. So think about the experiment on the right here. So here we have an experiment, 3 different replicants in 2 different experimental groups. Right? So on the top 3 pots, we have 10 seeds that were watered with regular water, and we see that in the pot number 1 we have 9 out of 10 sprouted. In pot number 2 we have 6 out of 10 sprouted. In pot number 3 we have 7 out of 10 sprouted. Then on the bottom we have some pots that were watered with Brondo, and on the left we have 9 out of 10 who was sprouted, 3 out of 10 in the middle, and then 5 out of 10 on the right. Now think about this data. None of these numbers is exactly the same. Right? Between these 2 sets we have one of the Brondo samples that has as many seeds that were sprouted as the highest number of sprouts in the water group. But if we look at these sets as a whole, we can think about the average of these numbers, and I have graphed the average of these 3 numbers on the bar graph on the bottom. But then we can use statistical measurements such as the standard error of the mean. So the standard error of the mean is, we're not going to go into the math, but it essentially measures how different each one of the individual measurements is from the mean or the average of these samples. So you can tell just by looking at the bottom row, there's actually more variation in some of these pots than there are up at the top. Right? But at the same time, so even though the average of these 2 groups is different, right? Because we have variation within them, how confident do you think we can be that these 2 numbers, the true inherent meaning of these numbers is different from each other? That's what the standard error of the mean, and that's what these error bars are meant to try and help us get a handle on. And we're going to think more carefully about how statistics and what they mean as we read this paper.